Let's go. Next is the alum company, DeepGenomics. Brendan, welcome.
Right.
We'll cue the video. Is there a video or is it?
So no video.
Okay.
Sergey Brin once said that videos of the early days of DeepGenomics would have been, of Google would have been a disaster. Just a big empty white screen with a cursor blinking in the middle. So what I'm going to do is try to kind of rein in expectations a little bit. I'm going to talk, like the last speaker, I really like the way he talked about a major problem facing us. And that's really what I want to focus on is the major problem. So in his case, it was the farmer being able to listen to the plant. And the goal of DeepGenomics is to enable each and every one of you to be able to act upon the information stored in your genome. And for that to be true for everyone in the world. And really there's a problem, which is that even though it'll soon cost less than a trip to the grocery store for you to obtain your genome sequence, currently we really don't know what to do with that information. There's not much we can tell you about how you should act on it. And it's quite tragic, really. So I call this the genotype-phenotype gap. And what I'm going to talk about right now is sort of the, if you like, the business opportunity and also enabling technologies which are going to change medicine drastically as well. In 20 years, you won't really recognize what's going on in medicine compared to today. So first of all, just a few numbers. 65%, think of your loved ones, friends and family. 65% of them will face a serious genetic disorder in their lifetime. 8 million births per year with a genetic defect. And I don't like to put money, dollar signs on this, but this costs about $5 million per child in the US healthcare system. Putting this market in perspective, the healthcare budgets of the US and Europe is 7 trillion per year. The global IT market is 3.7 trillion. And if you combine Apple, Microsoft, Google, and Amazon, it's only 0.53 trillion. So the problem is economically big, and it's also very big for us, of course, emotionally, thinking about our loved ones. So how about big pharma? Is big pharma going to solve the problem, give us new diagnostics and new drugs? This plot here shows the internal rate of return for the big pharma industry dating back to 1990. Back in 1990, the internal rate of return was almost 30%. That's pretty good. Put your money in big pharma. Recently, in 2012, the IRR of big pharma fell below the borrowing rate. And it's expected to go to 0 by 2020. Okay. That's a rather large elephant in the room. All right, so let's go back to genetics and really the reason DeepGenomics exists. So the genome was sequenced almost 15 years ago now. That's a lot of time. 15 years ago for the first time. And Eric Lander, who is involved in the genome sequencing project, pointed out, he quipped, Genome bought the book hard to read. And you know, it was funny at the time. It's starting to be not so funny. It's 15 years and a lot of money has been spent and more genomes are being sequenced, but we really still can't read the text of the genome. And if you ask the question why, there's various reasons, but it really comes down to an informational problem, how to make sense of the information in the genome. So I'm going to very quickly give you a tutorial on the genome. It's a bunch of letters, 3 billion letters in your genome. Each gene consists of a region called the promoter, the part that's shown in blue there, and then there's introns and exons. Essentially, the promoter contains information that tells the cell when to turn the gene on and off, so when to use that gene. And then these introns and exons, they get spliced together and cut apart to form proteins that are going to actually do things inside of your cell. So you can think of it like a book, and these introns and these exons and the promoters are like words in the book. Now, the current technology that's widely used for trying to understand the genome is to line up the genomes of people that are sick, say with cancer, line up the genomes of people that are healthy, and then look for individual letters that are enriched in the people that are sick. That's called a genome-wide association study. I kid you not. That's the typical technique that's used at a company like 23andMe, for example. It would be like trying to teach your child to read or trying to understand which books they like to read by just looking at the frequencies of letters in the books they like to read versus the ones they don't like to read. It doesn't make any sense. So the approach that we take at DeepGenomics is one based on biology, which is to understand the biological mechanisms that lead from your DNA or your genome to the proteins that play such a crucial role. I'm not going to say a lot about this. You won't be surprised to hear that it's complex, multi-layered. There are a lot of feedback loops, many variables, many hidden variables, many confounding variables. So it's difficult to work with. However, it is a perfect match to AI. Previously, people have tried to use, for example, rule-based systems. They just program if-then statements into computers. Didn't work. People have tried to use molecular simulation, where you write down differential equations describing molecular concentrations. Didn't work. So why AI? Well, intuitively speaking, biology is complex, multi-layered, and in the last 5 years, we've seen an exponential growth in the number of data sets that allow us to peer inside of cells and measure what's going on. So that's the key. That's an enabling technology. Different kinds of biotechnologies that allow us to measure large amounts of data. Now, the other technology, of course, is one that everybody in the room is familiar with, which is AI, the ability to make sense of that data. I just wanted to give you a quick glimpse into the kinds of data we have available. So on the x-axis here is the average gigabytes of data, so the size of the data set. And on the y-axis is the number of data sets. Now, if you look at genome sequencing data, that's DNA, there are roughly 10 gigabytes per data set. And recently, the number of such data sets went above 1 million. So that's a lot of data. But equally as important as DNA is all these other data sets, targeted RNA data sets and RNA-seq or RNA data sets that are measuring what's going on inside of cells. So I just wanted to give you a sense that in biology, there are now huge data sets that we can process using AI. So just in the last few minutes, I'll talk about deep genomics and how we're approaching this opportunity, this business opportunity, this opportunity to really change the world and improve human health, and using these enabling technologies. To give you a glimpse into our first market, if you like, our first, the first problem we're going to crack, I'm going to show you a specific example of spinal muscular atrophy. How many people know what spinal muscular atrophy or Spinraza? Just raise your hands. Right. Those statistics make sense. So spinal muscular atrophy afflicts 1 in 10,000 births. Previously, the child would usually die within the first year. Very serious disorder. In 2016, a new drug was approved called Spinraza. And I'm going to show you how it works digitally in terms of how the genome works. So there's an intron, there's an exon, and this is the gene that encodes a protein called SMA, or the spinal muscular atrophy gene. People often name genes after the disease that the gene is linked to. So there's an exon, there's a mutation in that exon. A C is converted to a T, and that mutation causes something to go wrong. Now you might think, okay, gene editing, let's just fix that mutation. Gene editing doesn't work that way, I'm sorry. But here's how the drug works. The drug is what we call a digital medicine. And if we just go downstream of that exon and look at the intron, the medicine binds complementary, so G's bind with A's, G's bind with C's, A's binds with T. It binds to this region in the intron, modulates a code word, if you like, a control statement that's embedded in the intron, and fixes the problem, okay? So I wanted to show you this example because it tells you that there's a combinatorics to this problem. It's not a sort of a simple, here's a mutation, let's fix it. Biology is complex. There are combinatorics. You need systems that can deal with those combinatorics, such as AI systems. So how long did it take to develop Spinraza? A long time. Here's the timeline. A lot of investigation into failed compounds, initial drugs, the final drug, the clinical trial, and so on. The goal of deep genomics is to take that timeline and squish it, make it really short, and also to be able to develop lots of drugs in parallel. And so that initial period of time there, where the mechanism and the initial compounds were explored, took 7 years. At Deep Genomics, we now have an automated system in place that allows us to scan for mutations, find mechanisms, and develop oligos within a few months. A couple comments on modularity. So when I talk about Deep Genomics and what makes us different from other companies that are bolting AI onto what they do, what's quite different about Deep Genomics is artificial intelligence is woven into the fabric of the company. Everybody in the company uses our artificial intelligence systems, and everything they produce, we're always asking the question, can that be used to improve our AI systems? Can we take the data and feed it back and make our machine learning and AI systems better? Really important to be thinking that way. That's how you get scale up. That's how you get a situation where your n plus first drug will be easier to develop than your nth drug to get a network effect on the technology side. So modularity, we think of modularity also in terms of traditional drug development, where you have your indication, you have your mechanism that you need to remediate, you have a compound that you need to design, you need to figure out whether that compound is going to have toxicity in mice and non-human primates, you need to put that into clinical trials and advance it as rapidly and successfully as possible through clinical trials. So all of those steps are modules at DeepGenomics, and we're building machine learning systems to accelerate all of those steps. Obviously, we test our compounds. We have a wet lab facility. We use cloud labs to scale this up, so it's always about scaling and generating more data and feeding it back. And one of the biggest things that we face in drug development is how to earn the trust of patients, their physicians, pathologists, ethic review boards at hospitals, insurance providers, and regulators. Okay, so if you're building a machine learning system and you're worried about trust, in medicine, those are all the stakeholders, and you need to earn their trust. We have ways of doing that. I'll just conclude by talking about our current 2-year goal. Our current 2-year goal is to use AI to search over 69 billion compounds, which is an in silico library that's just astronomical, much bigger than any library anyone's ever looked at, and identify 1,000 compounds that allow us to carefully and precisely control what's going on inside of human cells, and also to put 3 compounds into development. That would be IND-enabling work and ultimately clinical trials. Thank you.